Thank you for the opportunity to speak to you today. I'm going to talk a little bit about why honeybees are dying and the reasons that are behind these high mortality rates that have been observed since about 2007, and what in the university we're doing about it. And to start with, most of us, when we think about honeybees, with two things come to mind. One is bees produce honey, and the other is bees sting. One is sweet, one is not so sweet, the stinging part. But we should know that bees are much more than just honey and stings. Honeybees also produce and collect other high products. For example, they collect pollen, like in the second picture here seen to your right. You can see a yellow pellet, actually it's an orange pellet carried by a honeybee in her hind legs back to the hive. And we can collect that pollen for human or animal consumption. It's a product rich in nutrients like proteins and lipids. So they also produce beeswax, which we use in those wonderful candles that we might give as gifts in Christmas times. And they produce propolis, which is a natural antibiotic, by collecting resins from trees and adding some beeswax and saliva products into it. It becomes a natural antibiotic in the hive. But the most important thing honeybees do is they pollinate crops. That is, they transfer the pollen grains from the male parts, male organs of flowers, to the female organs of flowers to induce reproduction in those plants. And by doing that, they contribute to food production. In fact, about 1/3 of the food we consume in Western societies depends on the bee pollination services. So that's a lot, 1/3 of what we eat. Imagine if bees didn't exist. Here you can see a few fruits that were poorly pollinated by the wind or other means, not by honeybees. So they are reduced in size, or parts of it are developed, parts of the fruits are not developed. And that's because of lack of good pollination. When bees forage, that is when they fly out of the hive and visit flowers to collect nectar and pollen, the flower design forces the bee to touch the anthers. The anthers are the male organs of the flowers that contain the pollen, and the pollen contains the gametes, the male gametes, or male cells, reproductive cells of plants. And by doing that, before reaching the nectar, is the organs where the nectar is produced in the flowers. Bees carry these pollen grains to other flowers of the same species. They don't go to a different plant species. So they wouldn't carry pollen from one species to a different one. They carry pollen from one species to the same species. That's what we call fidelity. And that fidelity allows that plants reproduce and produce crops. So these are some of the reasons why honeybees are ideal pollinators. There are many pollinators, including other bees. There are about 20,000 bee species, but honeybees are ideal pollinators for a number of reasons, which include flower fidelity. I was talking about flower fidelity a minute ago. That is, they only carry pollen from one species to the same species. Then the need for pollen, I mean, bees use pollen as a source of proteins and lipids and vitamins and minerals to feed their larvae to produce new bees. So they are obliged to collect pollen, and by doing that, they help pollinate plants. They have plumose hair. That's a hair adapted to transport pollen, and it helps the bee to get the pollen attached to the hair until it reaches the female organs of another flower in which the bee um, deposits these pollen grains. And then the plant reproduces. They also live in populated colonies with lots of individuals. One colony of honeybees can have 50, 60,000 individuals. And those individuals, many of them, one third of them could be foraging and pollinating plants. This number is very difficult to reach by other pollinators. For example, bumblebees, you're talking about hundreds at the most, but not 20,000 individuals in one colony. And then they live in colonies that are um, housed in hives, which are these wooden boxes that can be managed and transported in trucks to different places where they're needed, and as many as they are needed. For example, here in Ontario, beekeepers uh, rent out their hives to pollinate blueberry crops in New Brunswick. About one third of the colonies that exist in the province, and that's about 30,000, 40,000 colonies depending on the year, are rented out for pollination services to blueberry producers in the Maritimes. And so you can transport as many hives as needed to those orchards and provide the pollinating services required by those orchards, and something you cannot do with other type of pollinators. When bees forage, as I said, they flower design. Oh, sorry, I'm going back. Um, these are important statistics for you to have an idea of the value of honeybees for food production. In Canada, it is estimated that the value of the pollinating services of honeybees is about $1.5 billion a year. We're talking US dollars. In the US, it's 14 billion US dollars a year. In the world, more than 150 billion US dollars a year. And the value of pollination services relative to the value of honey is about 20 times higher. And so in addition to this value, in Canada, 1.5 billion US dollars, you have to add another $8 billion of for the value of honey. Uh, More statistics regarding the value of bees, honeybees as pollinators in Canada. More than 300,000 colonies pollinate canola in the west of Canada. And Canada ranks number one for canola production in the world. And as I said, 35,000 colonies pollinate blueberries, mainly from here from Ontario. And Canada ranks number two for blueberry production just after the United States. So these crops require a lot of pollination by honeybees. And some of the reasons why we are within the first world producers of these products is thanks to the pollinating services of honeybees. There are other crops that benefit from bee pollination, including apples, pears, cherries, etc. Vegetable crops. Here are some examples: fruit trees. To the left, almonds, which is a crop that demands lots of pollination. In California, for example, more than 1.5 million hives are used every year to pollinate almonds. And the demand for hive pollination is increasing year by year because The consumption of almonds in the world, it's skyrocketing. Avocados in the center, crops that are produced in southern United States or Mexico require pollination from honeybees. Apples, vegetable crops like cucumbers, um, melons, watermelons, squash, etc. Berries, we produce lots of berries. We talked about blueberries in the center, but raspberries as well, and other berries are benefited by honeybee pollination. Seed crops, sunflower, canola, alfalfa. So lots of benefits from bee pollination, but bees are also valuable for other reasons. They teach us about ourselves. For example, did you know that we are about 15% bees? Yes. Genes associated to characteristics of human interest, like aggressiveness, immunity to diseases, alcoholism, etc., are present in the bees. There are ortholog genes in the bees. As you know, the sequence, the honeybee genome, as well as the human genome, have been sequenced. And when you compare the genomes, you find about 15% of the genes are orthologous. That means they have a common gene ancestor. And so, in a way, we are 15% bees. And we can study all these problems of human interest in bees that are easy to manipulate and work with instead of humans or larger animals. And we can learn about aggressiveness, we can learn about immunity, we can learn about how social behavior evolved. And bees are also for fun because it's easy to keep them in hives and they're great for hobby beekeepers. Many people are tired of working in enclosed environments, like in an office, and want to have some contact with nature. And beekeeping it's a good hobby for people who want to have more contact with nature and the environment outside of an office and find beekeeping as a great thing to do during weekends. And as a matter of fact, about 80% of our beekeepers in Ontario and across Canada are hobby beekeepers. The rest are commercial beekeepers, only 20%, although most of the hives are in the hands of commercial beekeepers. So there are many reasons to keep honeybees alive and healthy, even for selfish reasons. But we have a problem. Honeybee colony losses in Canada and around the world are constant since, as I mentioned, since about 2007. Colony mortality rates about 30% during the last 7 winters in Canada have been well documented. And the estimated losses in the value of colonies, the value of the honey yields that are lost, the value of the pollinating services that are lost and result in loss of crops is about $400 million annually just in Canada alone due to these losses. Here I can show you a graph of 2 bars showing the rate of mortality, colony mortality every year in Canada for honeybee colonies. And you can see 2 bars there, the yellow bar and the red bar. The red bar is the average colony loss for the entire country of Canada. And the orange bar corresponds to the specific losses in Ontario. And if you compare the red and the yellow bar when it was possible, before 2007, it was not possible because we didn't have records for the country, only for the province. And as you can see here, in most years, the yellow bar exceeds the red bar in 2014, for example, we lost nearly 60% of our colonies in a single year. Can't imagine what type of losses that if it occurred in other animals. We have about 100,000 colonies here in Ontario, and to lose 60,000 in a single year was devastating for the beekeeping industry. Anyways, beekeepers are resilient, and every year they're able to make up for their losses. And as a matter of fact, the number of colonies in the province and in the country has remained stable, and it has increased a little bit. In part because the beekeepers split the colonies that survive to make up for their losses, and or purchase imported packages of bees that come mainly from Australia and New Zealand. And that's how they refill the empty boxes where colonies die. So, what causes these high colony mortality rates? There are factors working in synergy, more than one. And most of us researchers agree on what factors are involved. What is still a matter of debate is the relative weight of each factor, which is difficult to answer. What of the factors that we know kill honeybees have more weight or have more importance or have more to do with these losses? But we have conducted our own research here at the University of Guelph. And to make a long story short, we follow the fate of hundreds of colonies in the province and studied different factors that could be involved in these losses. And the main conclusion of that study was the Varroa destructor, which is a mite like a tick that lives and feeds upon lives on the honeybees and feeds upon its hemolymph, which is equivalent to its blood. It's the main culprit of winter colony mortality and reduced populations of honeybees. 85% of the deaths are related to high infestation levels of this mite. We also found that a fungi, a couple of fungi, Nosema species, Nosema serrani and Nosema apis, cause infections, and these infections are one of the leading causes of poor colony build-up in the spring. So colonies don't die out of Nosema infection in the winter, but don't develop, don't grow in population, and therefore they wouldn't produce as much honey or pollinate as much. And then the third major factor that we found associated to colony mortality were pesticides, mainly neonicotinoid insecticides, which are molecules similar to nicotine, and are the main insecticides used worldwide in the last 2 decades. And it was a major factor associated to bee losses in spring. Therefore, our research focuses on Varroa mite control, on viruses associated to Varroa, because Varroa transmits a number of viruses that are damaging to the bees. We also work on Nosema disease studies, those caused by the fungi Nosema species. We have been studying in the last decade the effect of parasites on immunity and behavior, how Varroa, how Nosema affect the immunity and the behavior of honeybees. And we have also studied the effect of neonicotinoid insecticides on and pathogens, Varroa and Nosema, on bee health. Because as we said, it's not a single factor, it's this synergic effect of several factors. So bees could be affected simultaneously by pesticides and by parasites and pathogens. So the Honeybee Research Center, HBRC, Honeybee Research Center. So we have a research center here at the University of Guelph that has been traditionally been involved in apiculture programs since 1894. Very few universities can have the pride to say that they've been doing research and teaching apiculture for more than 100 years as we have. And so in this Honeybee Research Center, we not only conduct research, but teach more than 500 students a year, teach and train new scientists, train uh, um, beekeepers that are in the community. And we also educate in the community. We have visiting programs at the Honeybee Research Center. This is the Honeybee Research Center, one of the main buildings of it. It's the Townsend House seen from the air down there. You can see some of the hives down there. It's a close-up of the hives here. Some are still wrapped on black plastic wrapped on the back. That's how we protect them from cold in the wintertime. And then in spring, we start removing those wrappings to allow the bees to grow and start foraging. We also have at the Honeybee Research Center a molecular and pathology laboratory where we conduct studies. And as I said, one of the subjects of study is the varroa mite. Here's a picture of it. It looks like a tiny crab. You can see the reddish little crab on a pupa on one of these pictures, or on an adult bee in the picture to the right below. And it's a parasite that feeds upon the blood and fat tissue of honeybees. They pierce the cuticle of the bee and sucks the blood. So if you can make a fist and imagine to have a parasite the size of your fist feeding upon your blood, you can have a comparison of how big this parasite is for the honeybee. For our naked eye, it's small, the size of a pinhead, but for the bee, it's huge. You can see here a picture of an bee on the top that was not parasitized by varroa, and a picture of a bee below that was parasitized by varroa inside the cell, and it emerged smaller in size with damaged wings. And of course, those bees don't live too long. How do beekeepers control the varroa mite? Well, they use synthetic miticides, which are chemicals, pesticides, that are effective for some time, but after 5-10 years, mites develop resistance to the chemical ingredients in these compounds. They're also toxic to the bees, and they may contaminate honey and wax, which is supposed to be a healthy product for human consumption. So we need alternatives to these synthetic miticides, and we have tried biological control of varroa mites by using fungi that were able to kill mites, but unfortunately they were also able to kill bees, and therefore were not a suitable solution for mite control out of the miticides, synthetic miticides that we use. So we have also tested natural compounds, a number of them. Here is a list of them: methyl salicylate, menthol, camphor, carbacrol, clove oil, etc., etc. And to make a long story short, we tested many of them until we reached a few that were very effective. And the effectiveness of those compounds was related to the formulation, that is the method of delivery into the hive. And we came up with new formulations of essential oils or components of essential oils. For example, thymol, it's a component of the essential oil of thyme, which is a plant that we use in kitchens. And it was developed here at the Honeybee Research Center, highly effective. Our trials in Ontario yielded more than 90% mite control. Then colleagues in Alberta tested it independently from us and got an even better mite control, 95%. We also have found an effective formulation using oregano oil, which is also used in kitchens, and 85% mite control. So we're happy with them, and now the Ontario Beekeepers Association is seeking registration for one of our formulations. That's not our job. We don't commercialize these products, but the OBA, the Ontario Beekeepers Association, may. And we have also studied other ways of controlling the mite. For example, trying to develop resistance, genetic resistance in the bees against the mites. And by studying these behaviors, we have focused focus on two mechanisms of resistance, which are two behaviors that provide resistance to the bees against the mites. One is the so-called grooming behavior, the ability of the bees to remove mites from their bodies, either individually or with the help of other nestmates that groom among themselves. And then hygienic behavior, which is the ability of the bees to recognize cap cells where pupae are developing, and in which the mites are present and are reproducing. So bees are able to detect those cells even beneath the capping, the wax capping, and open the wax capping and remove the cells and kill the mites sometimes. So here is a bee that was tested for grooming behavior. We place a little mite, a varroa mite, on her body. You can see it probably here, and it's on her thorax here. And she's making these grooming movements with her legs, trying to get rid of the mite from her thorax. And I would say this is intense grooming behavior because she's using more than two legs to try to get rid of the mite. And eventually she drops the mite on the floor. Sometimes they bite the mite and get rid of it. And so we're trying to select bees that express this behavior at a higher frequency and see if it's a selectable character. Curiously enough, this behavior, grooming, it's influenced by a gene called neurexin, which is also present in mammals. In humans, this neurexin is related to autism. In mice, it's related to grooming behavior, just like in the honeybee. And so we're trying to come up with a lab technique to use this gene as a genetic marker for assisted marker selection. We study honeybee viruses that are transmitted by varroa mites, for example, the deformed wing virus, the Israeli acute paralysis virus, and the acute bee paralysis virus. Here you can see a picture of a honeybee with crumpled wings. They are completely damaged, and that's because of infection by the deformed wing virus, one of the most pathogenic viruses transmitted by the varroa mites. And that's another reason why you want to control the varroa mite so that it doesn't transmit these deadly viruses. Nosema disease, it's an intestinal disease of the honeybee, which is caused by two species of fungi, Nosema apis and Nosema cedrani. And it's fungus that forms spores that contaminate the comb in the hive. And when the spring comes, the newly emerged bees, young bees, clean the cells with their tongues so that the queen can lay eggs in them. And by doing that, they acquire the spores that are on the combs and ingest the spores. And when they reach the digestive system, the spores germinate and invade epithelial cells in the midgut of the honeybee and cause a series of symptoms, digestive symptoms, causing them not to absorb nutrients and weaken the bee and shorten their lifespan. Here you can see a couple of pictures showing the spores of Nosema. On the top left, it's just a contrast phase microscope in which you can see those spores. I can show you one here. I can show you one here. And in the picture below, the spores are stained with a fluorescent dye, and that makes them glow, and they are easier to detect. But the fact that you see spores under a microscope doesn't tell you what species you're dealing with. And it is important to find out what species you're dealing with, because Nosema cerani seems to be more pathogenic than Nosema apis. And for doing that, we have a PCR technique. And here is a photograph of a gel showing bands of a PCR reaction, in which you can see a band up here that it's a fragment of DNA from Nosema apis, 300 base pairs, and then another fragment here below, it's for Nosema cerani. And we use a B gene, which should always be present in the reaction, as a control. I mean, if this B gene band is not shown, that means the reaction had a problem, and not, we cannot give it as negative. So, but if we see the fragment for the B gene here and not bands for Nosema apis or cerani, you can give the sample for negative. So we inoculate bees with Nosema spores. Here you can see this photograph to your left, in which a bee is held by the wings, and it's given sugar zero containing a known amount of spores of Nosema, Nosema cerani in this case. And then the bees are placed in cages like the ones you see to the right, and the picture in the right. These cages are kept in an incubator, and we can study many things, longevity of the bees, behavior, etc. We have studied how these parasites, both Nosema and Varroa, affect the immune system of the bees, and how bees can use their immune responses to defend themselves naturally against parasites. And we have studied ways of increasing the expression of these immune responses to parasites. For example, we have given them compounds that stimulate their immune system. It's like vaccinating the bees in a way, to try to make them respond more efficiently against pests and parasites, and be able to defend themselves. The other major stressor of honeybees that we encounter associated to colony losses is pesticides. And this has been a major problem for many years. 25% of the known pesticides are highly toxic to bees. But today, the main issue is with the so-called neonicotinoid insecticides, which are the most widely used pesticides worldwide. These are systemic insecticides, that is, they spread throughout the tissues of the plant when they grow. Here you can see a picture of seeds treated with neonicotinoids. The neonicotinoids mostly used are imidacloprid, thiamethoxam, clothianidin. And these seeds are colored just to show, tell the grower that those seeds have been coated with neonicotinoid insecticide. So when these seeds are planted and the plant grows, all the tissues of the plant carry the pesticides, the neonicotinoid pesticides. And when a pest eats up the plant, gets poisoned and dies. Unfortunately, visits by bees to the plants to collect nectar or to collect pollen, they also collect pesticides from them, or they are exposed to the pesticides when the plants are sprayed, or well when the seeds are planted. Here in Ontario, the main problem was with when the seeds were planted for corn and soybean, for example. Here, on the top left, there's a picture of a tractor with a pneumatic planter here. When the corn seed or the soybean seed is planted, these pneumatic machines release this powder. What is this powder? This powder, it's a talc that is used as a lubricant, and this lubricant is used in the injectors of the machine so that the seeds can travel faster through the injectors and are injected into the soil. But much of this dust becomes contaminated with the neonicotinoids, and then it's released in the air and drifts away and lands, like in the picture to the right, for example, on dandelions or other types of flowers visited by bees, or in puddles, like in the picture on the bottom, etc. So bees visit these places and become intoxicated with the pesticides, and many of them die. As a matter of fact, in 2012, 2013, we had major spring bee losses, more than 200 incidents of acute toxicity were reported that those years, which involved thousands of colonies, which is the largest number of incidents of toxicity by neonicotinoids worldwide to date. When PMRA, an agency from Health Canada, analyzed samples of dead bees from these fields, they found that 70% of the bee samples were contaminated with high levels of neonicotinoid insecticides. So it was a smoking gun right there. So, but the argument that is made by some growers and the chemical companies that produce these pesticides is that the amounts of neonicotinoids found in nectar and pollen of plants which grow from seeds coated with the pesticides is so small, so little that they wouldn't have harmful effect on the bees. So we have conducted studies funded by OMAFRA, the Ontario Ministry of Agriculture, Food and Rural Affairs, to look at potential synergic effects of not only pesticides, but also pathogens and pests together with the pesticides, and look at the effects of these pesticides alone or in combination with pathogens on the susceptibility of bees to diseases, their ability to learn, their ability to survive, and study their behavior. For example, we have studied bees that were treated with sublethal doses of these pesticides equivalent to those that they would find in the field, and found that they affected several of their behaviors: grooming behavior, foraging behavior, hygienic behavior, and the expression of many genes associated to bee health in general. So we have also studied how bees express this immune resistance against this pathogen, whatever, and ways of increasing immune responses to the parasites. I mentioned that we give them some immune inducers to increase their resistance to pathogens. Many people ask us, how can we help the bees? And some of the things that you can do as the general public to help the bees is eat locally produced honey. If you eat locally produced honey, that is honey produced by a local beekeeper, you're helping the bees. Why? Because beekeepers are the only one who can keep those bees alive. Beekeepers are the one who split the colonies, are the one who raise queens to produce bees, are the ones to keep them fed, are the ones to grow the colony. So if we don't have beekeepers, we don't have honeybees. at least in commercial settings or in hobby settings. So if you eat locally produced honey, you keep a healthy economy in the beekeeping industry, and by doing that, you're helping produce more bees that are needed for the pollinating services that plant requires. Plant flowers for bees, trees, and plants. We have produced a leaflet here at the Honeybee Research Center with examples of some plants that could be planted in your gardens as a source of nectar or pollen for the bees. Obviously, don't spray roadsides, don't spread dangerous pesticides like neonicotinoids in your gardens. If you have land, allow and grow vegetables or grow other crops in them. Allow part of your land to naturalize. Let the weeds grow, because if the weeds grow, they provide food not only for the honeybees, but also for local native bees and other native pollinators. And of course, support research on honeybee health, as we have a large community supporting our efforts here at the University of Guelph, and we're very thankful for that. So there's lots to do to keep those bees healthy and alive. Finally, I want to recognize the many people in our lab. I just have uh, the honor to lead them, but we have other scientists like Dr. Goodwin who help us a lot. He's a molecular biologist here at the School of Environmental Sciences and has partnered with us in some of the projects that we study. Paul Kelly, our uh, beekeeping and research manager at the Honeybee Research Center, and a number of postdocs and graduate students that are named here. We also have in visiting students and scholars from many countries that are trained here and participate in the different experiments that we conduct here at the Honeybee Research Center. So that's all I have to say for today. I thank you very much for listening to my talk, and don't thank me for my talk. Thank Honeybee.